All right, so after the DNA replicates, right? So if we're thinking of the cell life cycle, so there was G1 for growth, then there was S for DNA synthesis, and G2 for more growth, and now we're in mitosis. Okay, so mitosis, specifically what it is, it's pretty simple. It's separating those replicated DNA molecules, okay, those sister chromatids, so that one goes to one side, one goes to the other, and then we're going to get two new cells, identical to each other, identical to the parent cell. Okay, so it happens in our bodies, okay? It says 4, replace worn out old cells with fresh new duplicates, okay? So not only in our bodies, they show this picture, this guy cuts himself. How are things going to fix themselves? Through mitosis, so growth and repair. Here, look at this. Here's this seedling, and it sprouts and becomes this plant. And at the root, okay, and we're going to look at this in lab. At the root, there's a lot of mitosis, a lot of growth, okay? So it says most cells are not immortal. Mitosis generates replacements. Yes. So this happens in our body. This happens in plant cells also for growth and repair. So, oh yes, there is a definite programmed cell death. Apoptosis is this pre-planned process of cell suicide. Certain cells are targeted for that. So they do that. Some cells do that. Here are red blood cells. Red blood cells are actually made in the bone marrow, and there are stem cells in the bone marrow. They're made, and the cells circulate in the blood, but these ones that you see on your screen, they don't replicate. They die. Okay, every like 3 months they die, and then more. The stem cells make more fresh red blood cells. So, mitosis enables existing cells to generate new genetically identical cells. This makes it possible for organisms to grow and to replace cells that die. So, what's happening? It's actually super duper easy, okay? It's just the words that get in the way. The process is easy, okay? If we don't use any words, okay? Here's the parent cell. And if you look, bam, okay? Sliding, making 2 daughter cells, okay? I don't know if I can move. Yeah, I'm going to move myself up here, okay? So here is the parent cell making 2 daughter cells. That's easy. Yeah, there it is, okay? Now, again, the daughter cells are identical to each other, identical to the parent. Now, what gets difficult are all the words, okay? So if you remember, in the S phase, the chromosomes have replicated, and the chromosomes are sister chromatids. And if we look, okay, so if you look here, there's a dark blue, light blue, dark orange, light orange. And then they've replicated, so that's why you see kind of an X. But it's like there's one chromosome, there's the other, they've replicated, they've gone through the S phase. Here's the light blue, dark green, light green, da-da. And then those replicated chromosomes get in the middle of the cell. We call this the equator. Then it's those sister chromatids, those replicated chromosomes that are moving towards opposite sides, opposite poles. And then we have one cell with the chromosomes and one cell. And if you look, there's one dark blue here for this cell. There's one dark blue here for this cell, chromosome. One light orange, one light orange, one dark orange, etc. So this is the overview of mitosis. Now what happens is there are, quote, stages or phases of mitosis, and so there are names for them. So when we start seeing the chromosomes, it's prophase. When the chromosomes are lined up in the middle, think of middle, meta. It's metaphase, middle, meta. Then the chromosomes made out of the sister chromatids are separating towards opposite sides, anaphase. And then we start to see 2 new cells, that's telophase. And then 2 new cells, there it is, okay? So again, I'm gonna have videos on this, and I do want you to look at it. It really helps to see a video Also in lab, we're going to look at cells that are actually undergoing this. We can look at them under the microscope. Beautiful picture, yes, of cells that have just replicated. You're like, Miss Collins, there's nothing beautiful about this. Oh, you wait and see when we get into lab. Okay. So again, the chromosomes have had to replicate before mitosis. So here's the chromosome replicated. These are sister chromatids. Yes. There's what's called the spindle fiber. There are these protein filaments that are going to help separate the sister chromatids from one another. Details. Okay. So in the cell cycle, the majority, the G1, SG2, that's called interphase. Okay. Interphase. So it's just a cell doing its thing. Okay. Then we're going to start mitosis. When we start mitosis, if you look, here's the cell membrane. Here's the nucleus, and the DNA are in there. Right there, you can't see the DNA, it just looks dark, okay? But when mitosis is going to happen, the chromosomes start to condense, and they look like little fibers, and you can actually see that. You see that under the microscope. Again, we're going to look at that in lab. So that is called prophase. There are distinct sections of prophase, and I will ask this on the test, so you need to make sure you have this down. In prophase, the two things that I'm going to ask you, the chromosomes condense, right? So they become visible, right? It says sister chromatids condense, okay? The other thing, the cell, the nucleus, the nuclear membrane starts to break apart. Nuclear membrane breaks down. So those two things are the signals of prophase. Okay, meta, again, what did I say? Meta, think, middle, okay? So the sister chromatids, the chromosomes made out of sister chromatids are lined up in the middle, yes. Anaphase, the sister chromatids are being split from each other, moving towards opposite sides of the cell, and we can start seeing that. This is the spindle fiber, the protein filaments that actually attach to the chromosomes and pull them apart. So it says sister chromatids, pair pulled apart by the spindle fibers. One full set of chromosomes goes to one side, the other goes to the other. And then this is called a cleavage furrow. We're starting to see two new cells. And again, you can see this really well. If I say how many chromosomes are going to end up in this cell and how many are going to end up in this cell. So 1, 2, 3, 4, 5, 6 in this cell. 1, 2, 3, 4, 5, 6 in this cell. And if you look, long blue, short orange, dark green, long light blue, dark orange, dark yellow. Bam! They're identical, okay? And we're starting to see two new cells, so this is telophase. Chromosomes begin to uncoil as the nuclear membrane reassembles, the cell begins to pinch. There's a last section. Oh, it doesn't show it in the book. There's a last section where the cytoplasm itself and all the organelles in the cytoplasm split. That's called cytokinesis. Okay, cyto means cell, kinesis, it's movement. That's it, okay? Again, I'm going to have some videos for you. I'm going to have some sites. Please click on them and look, okay? All right, let's continue. That was the end of mitosis, okay? So again, this is beautiful because it's an overview, right? Shows everything. And then here shows when the sister chromatids are made, right? They're condensed, they're replicated, and then the different phases. So interphase, most of the cell cycle, the cell's in interphase. So here is prophase, here's metaphase, here's anaphase, and here's telophase. Okay, great. Now, what if things don't go right? Yeah. What if things don't divide the way they're supposed to? What if they keep dividing? Okay. Quote, normal cells have those checkpoints, right? And they stop dividing when they're supposed to stop dividing. Well, cancer cells lose that signal, right? They don't stop dividing. The word cancer, okay, cancer. If think about astrology, cancer sign is what? It's a crab, okay? So it's uncontrolled cell growths. Usually we think of tumors. The tumor looks like it's spreading, okay? And what's really bad, okay, so if someone has like some cells and they're localized, well, you can, and if they're having problems, you can cut them out. Okay, with cancer, what's so dangerous about cancer is those cells can, the word is metastasize. They can leave that site and go other places. And when that happens, now it's much harder to get those cells out of the body. And cancer is called the intimate enemy. Intimate because it's from that person's body and it's causing problems. And you're like, what's the problem? It's that person's own cells. What's the problem? Well, the cancer cells, the tumor cells are growing and they grow fast and they take nutrients away from the healthy cells. If they take nutrients away from the healthy cells, they're going to kill the healthy cells. And then when you hear, one of the signs of cancer is the person gets super skinny. The reason they get super skinny is the tumor cells are taking nutrients away from the healthy cells and the person loses weight. Again, you're like, what's the big deal? Well, they kill the healthy cells because they're taking the nutrients away. And then the cancer cells don't have a function and the organs start to shut down. So it's a horrible, horrible disease. People, I'm going to say this. I don't want you to get scared, okay? The statistics are 50% of the population is going to have cancer. So that's 1 in 2 people. Now, we have come a long way with cancer in the sense that a lot of cancers are very treatable. So that's a good thing. Yeah. And maybe hopefully within our lifetime, like all cancer will be treatable or curable. There are some cancers that people died of, like breast cancer, that used to be a death sentence. It's no longer a death sentence for a lot of people. There's colon cancer, there's prostate cancer, lung cancer, there's all kinds of cancer, right? So it's where it originates. Okay. So here it says cell division out of control may result in cancer. What is it? What is cancer? Unrestrained cell growth and division can damage adjacent tissues. Some can metastasize. That word metastasize is when they leave and go set up shop somewhere else. Can cause serious health problems and is the second leading cause of death in the United States. So the first leading cause of death is cardiovascular. The second is cancer. So what is it about the cells? Okay, there are 3 things cancer cells do that normal cells do not do. Normal cells, when they're growing, if they hit other cells, that signals them to stop growing. Cancer cells don't do that. They just keep growing and it makes that mound, okay? It says cancer cells have no contact inhibition, okay? So it says cancer cells have several features that distinguish them from normal cells. No contact inhibition. Cancer cells ignore the signal, continue to divide, pile up on one another. That's one thing, okay? Another thing. Cancer cells divide indefinitely. Normal cells, they have those telomeres and then they stop. They eventually stop dividing. Cancer cells don't have that. They just keep dividing indefinitely. Again, that sounds good, except for these cells have no function other than to take nutrients away from the healthy cells, so that's what's bad. And then the last thing, this is the really bad one. Cancer cells have reduced stickiness. So normal cells stay in the region they were made. Cancer cells, that word malignant, right, and the word metastasize, cancer cells take off, okay? They can go into the blood, they can go into the lymph. And if you've ever heard of someone having, let's say, breast cancer, and they treat it, and they cut the cancer out, they might radiate the tissue, and then a little later, maybe a few years later, you hear they have lung cancer or they have brain cancer. It could be the same cells. It's just the cells left and now they're setting shop somewhere else. Okay, we're going to go ahead and stop here and continue.